welcome to everybody who is joining us. Today, we will be going through our R&D portfolio and exposing that to you. So there will be no new financial nor trading information disclosed today. And here you have the standard disclaimer. We will be publishing our results for the half year ending September towards the end of this month, November. So brief reminder about our strategy. Three key planks to our strategy. Number one, which is what we're going to focus on today, is investing in R&D to develop new innovative products, focusing on swine and poultry and prevention of infective disease that causes economic challenges for the producers in that area. We also continue to create partnerships and make strategically and financially sensible and robust acquisitions to develop our core strengths. And we also continue to develop our core brand of Avicin into new geographies, new medical claims, and new species, as we recently did when we announced just a few months ago that we had successfully got the sow safety claim, as we might call it, which allows us to sell into gilts and breeding sows and removing a theoretical restraint in there. That allows us access into a new segment. We're also doing the same in poultry and have a zero egg withdrawal in some countries and pending in others. So that's the three planks of our strategy. Why are we in swine and poultry? Well, simply put, there are going to be very shortly 2 billion more mouths to feed in this world. And as the demographic trend of increasing human population multiplies with increasing household income, the net effect is the demand for a larger amount of high quality protein. And the vast majority of that will be supplied by poultry and swine meat. Poultry will become the #1 source of human protein and swine following very closely behind. And that demographic trend and following wind will be with us for a considerable period of time. So we are researching into areas of significant need and significant economic advantage. So there's three ways in which we can shape the future of swine and poultry prevention. First of all, we will present some vaccines against some of the key economic diseases in poultry and swine. In the first column, that's Mycoplasmas in poultry, and two diseases in swine, Mycoplasma pneumoniae and porcine circovirus. We also will bring the innovation of monoclonal antibodies into controlling PRRS, into controlling poultry disease, necrotic enteritis, and in some anti-parasitic conditions as well. This is important because monoclonal antibodies have revolutionized human health, particularly in the areas of oncology, and monoclonals have added billion-dollar medicines into the veterinary companion animal market, and ECO will bring this innovation to livestock. As part of that, and as a follow-on to some of our other innovations, we also have innovation in our delivery technologies. For example, reverse thermal technology, which simply means that we can extend the lifespan of our products in the animal. And we also, in swine and poultry, it's important to have mass delivery mechanisms. And we will be employing this technology where we can use friendly commensal bacteria to deliver the product to them in a mass market, a mass medication methodology. So making it easy for the producer to use the products that we are researching. We've got an agenda here that will run through, starting with Dr. Hafid Benchaoui, our head of R&D, a number of his team, and also an external speaker. We're very pleased to have Professor Naola Ferguson. And that will, the R&D presentations will be followed by financial valuation presentation from Chris Wilks. I'll come back for a summary, and then we'll progress into Q&A. So with that, I would like to pass over to Hafid, our head of R&D. Thank you, Hafid.
Thank you very much, David, and good afternoon, everyone. I'm very happy to be with you today to talk about R&D's vision and strategy, and also give you a progress update on our pipeline. Our efforts in R&D at ECO Animal Health are firmly focused on biotech innovation for swine and poultry medicine, more specifically infectious diseases across a spectrum of viral, bacterial, and parasitic diseases of economic as well as animal welfare importance that affect those two sectors of the animal health industry. Our ultimate aim is to provide breakthrough solutions that address the unmet needs of the pig and poultry producers, and at the same time create and build value for the business. Our goal is to develop novel vaccines and biologics and unique, effective, and sustainable alternatives to antibiotics. And in pursuit of that goal, and as you've heard from David, we have invested in the discovery and development of novel single-domain monoclonal antibodies, also known as nanobodies, and we have done that for both swine and poultry. We have filed a number of patents to date to protect our intellectual property in that space, and we have recently generated some very exciting and clinically relevant data with this technology, which the team will share with you today. What you see on the right-hand side of this slide are pictures of the contributing speakers who will be joining me today. And I'm delighted that our external guest speaker, Professor Naola Ferguson Noel, has agreed to join us this afternoon, really to set the scene with an introduction on a key area of interest for ECO. So Naola will talk about the importance of poultry mycoplasmas. Naola is a key opinion leader and a world-recognized expert in this area of poultry infectious diseases. She's also the driving force behind our two poultry mycoplasma vaccines, which are now in late-stage development. And those are key innovations that came out of a successful collaboration and partnership between ECO and the University of Georgia. Now, converting those vaccine assets from academic pursuits into registrable products is a complex and also a demanding endeavor that Natalie will talk about. So Natalie will walk you through the significant traction we have gained and the progress we have made in relation to the development and the regulatory submissions for those two vaccines. So Dr. Natalie Desloges is a global project leader. She's in charge of our late-stage development poultry vaccines. After that, we will hear from Brian. Dr. Brian Martinson will be joining us from the US, is our global project leader for the swine biologics portfolio. And Brian will give you an update on our swine bivalent vaccine and our first-in-class antiviral monoclonal antibody against PRRS virus. We're very excited about this breakthrough, and Brian will share with you for the first time some really promising clinical data that we have generated with this innovative and game-changing technology. We'll then hear from Alphonso. Dr. Alphonso Lopez is leading the charge on our novel injectable long-acting antimicrobial for the treatment of swine respiratory disease. And Alphonso will also talk to you about an innovation we're pursuing in gut health for poultry. This will be a biological solution and an alternative to the use of antibiotics to tackle a hugely important and costly enteric disease in broiler chickens, that is necrotic enteritis. And we'll finish the R&D presentations with Dr. Mike Huether, our senior consultant, who will join us also from the US, and he will give you an update on our vaccines manufacturing strategy, particularly in relation to our late-stage vaccines and how we are preparing to roll them out onto the market once we obtain regulatory approval. But before I turn things over to our guest speaker, I'd like to quickly give you an overview and an insight on how we do things in R&D at ECO Animal Health, our strategy, our operational model, and importantly, give you an update on the pipeline progression and the value we expect from each component of that pipeline. We typically reinvest around 10% of our revenues to support our end-to-end innovation efforts, which has amounted to 9.7 million pounds sterling for the current financial year, plus just over 800,000 on our new R&D lab, which I'll talk about shortly. Over the past 5 years, we have built a portfolio of intellectual property to secure our future exclusivity. And the substrate of those IP filings is, of course, the science and the technologies contained in our pipeline, which is balanced across the two sectors of interest, swine and poultry, and also across stages of development. One thing to point out here is that compared to 5 years ago, when we just started investing in innovation, our pipeline is maturing with more projects in clinical and late stage than in early stage. We are a lean operation that leverages external partnerships globally, and we ensure that our processes are fit for purpose, allowing our people to do good science with minimum red tape and maximum momentum, speed, and efficiency. And in line with our lean operational model, we have indeed very little infrastructure, and this helps us keep our fixed costs under control and to dedicate the vast majority of our funds to the projects. So our model is based on external partnerships and external innovation to bring in new ideas, new platform technologies, to design discovery programs and to execute those programs to generate data. Some of our process development is now done at our new R&D lab in the US. We feel this gives us flexibility and pace and allows us to move fast. After process development, our scale-up and manufacturing are then outsourced, and Mike will talk more about that later. So our vaccine development model is based on retaining the leadership and outsourcing the execution through our network of CROs or contract research organizations. In terms of speed to market, we prioritize the US registration, which opens up the door to all other geographies that want to see USDA approval before granting local approval. And then, of course, Europe, the UK, and China. China has its own unique requirements for registration and manufacture of vaccines, and we have designed a strategy to specifically deal with that market in the future, which Mike will touch on briefly later. I mentioned our new lab in the US. It is located in Des Moines, in the Midwest, and has been operational since April. We have 4 scientists working at the lab, and I truly believe this gives us nimbleness and speed, particularly for our mid and late stage projects and assets. And the reason for that is simply that there are things that require multiple iterations, trial and error, and refinement, and those things cannot be outsourced easily, efficiently, and cost effectively. Once our process parameters are optimized and well-defined, we can then more effectively outsource. Over the past 6 years, our strategy has been to shift the way we spend our R&D funds of 10% revenue from the lifecycle management of existing brands to funding true innovation and building value through a pipeline of vaccines and biologics for our 2 sectors of interest. It is also worth noting that when we move projects from early stage to mid and late stage, they require increasing levels of funding. For example, we only need a few milligrams of an active ingredient when we are at the discovery stage, when all our testing is done in the lab. As soon as we move to animal studies, we need to produce those materials in the gram scale. And as we advance further into full development and pre-registration, we are talking about the kilogram scale. That gradual scale up of production that happens for advancing projects, together with the animal efficacy and safety studies that we need to conduct, requires significant investment. When we set out to work on a new idea and a new product concept, there are three fundamental questions that we aim to address. Does the product work? Is it safe for the animal, the consumer, the user, and the environment? And can we make it consistently to the required scale and quality standards? This requires a rigorous process of stage gate transition from discovery in our in vitro systems to proof of concept in the diseased animals under controlled conditions, to exploratory development when we optimize the dose and the formulation and explore manufacturability at larger scale, to full development where we scale up production and take the candidate product to the field to generate real-world safety and efficacy data under typical conditions of use. And then ultimately submission of the data to the authorities for product approval. The concept of PTRS, or probability of technical and regulatory success, that you see on the left-hand side column, allows us to quantify uncertainty across time. As we make progress and generate data, we close the knowledge gap on those three questions I mentioned earlier: efficacy, safety, and quality. And we gain greater confidence in the product concept. It's a long and complex process, and all our projects go through that process, but we believe we have the know-how, the experience, and the agility to shorten the pathway and the time frame whenever we identify an opportunity to do so, without, of course, compromising the science or the regulatory compliance. Here we have used the imagery of the funnel to capture our pipeline and to categorize our assets into early stage, which is the discovery stage, where we validate the target and screen out the suboptimal candidates in the lab. The clinical stage is typically when we have successfully selected a lead candidate, scaled it up, and assessed its efficacy and safety in a clinically relevant setting of diseased animals. Late-stage assets, which are in development and/or regulatory submission. As indicated earlier, we have a balanced mix of poultry and swine projects, predominantly in the vaccines and biologics space, the exception being our long-acting antimicrobial, number 10 on this slide, which is a small molecule antibiotic. To de-risk the pipeline, our strategy has always been to adopt a mix of tried and trusted concepts, such as the poultry Mycoplasma vaccines, as well as disruptive innovations such as the monoclonal antibodies. The projects you will hear about today from the team are the ones that are circled, and they are in clinical stage or late stage. Focusing now on those clinical stage and late stage assets and excluding the early stage projects, this slide captures the value of our pipeline expressed as peak sales for each project as the bubbles in teal color. We have the development costs in green bubbles, and the rosettes are the timings of first approval for each asset. On aggregate, the projected value of our mid and late stage pipeline is about 213 million pounds worth of peak revenues. Revenue generation will start with our poultry Mycoplasma vaccines, Ecovaccine MS and Ecovaccine MG, with the anticipated US approvals in the first and second quarter of 2022. 2025. I'd like to conclude by saying that we have solid foundations to establish ECO as a key player in biotech innovation for swine and poultry. We have competent, experienced, and highly committed people. We have an active, dynamic, and well-equipped research team in our lab in the US, working in close collaboration with our network of contract development and manufacturing organizations. And we, of course, have an exciting, balanced, and maturing pipeline of proprietary innovations, which will start generating financial value within the 2025 horizon with our poultry mycoplasma vaccines. And that leads me now to hand over to Naola, who will talk about the importance of poultry mycoplasmas and the relevance of our upcoming vaccines to the poultry producers. Over to you, Naola.
Thank you. It's a pleasure to be with you all today to talk about avian mycoplasmosis, which is my area of specialty. So just a very brief introduction to Mycoplasma. It's not a bacteria that many people have heard about, but it is a very unique bacteria. Most bacteria have some kind of cell wall, gram-positive, gram-negative, but mycoplasmas do not have a cell wall, and it gives it some very unique properties. There are mycoplasma that infect just about every being on Earth that you can think about. There are mycoplasmas that infect insects, plants, as well as mammals and reptiles and everything like that. Mycoplasmas are considered to be the smallest free-living organisms, and they also have the smallest genome of any free-living organism. And a lot of the research and interest around mycoplasmas has revolved around that. I'd also like to mention that mycoplasmas are very host-specific. So the poultry mycoplasmas that we're going to talk about in the next couple of slides really only infect poultry. Gallinaceous birds in particular, which would be things like chickens, turkeys, pheasants, anything like that. So in terms of importance, the avian mycoplasmas, as I said, are very important to the poultry industry, and they tend to infect only poultry. So next slide. So one of the most important economically, as well as in terms of impact and virulence, avian mycoplasma species is going to be Mycoplasma gallisepticum, or MG as we call it. So all of these mycoplasmas in poultry have a global presence. Everywhere you have poultry production, everywhere anybody is trying to grow any kind of poultry, they have to consider and have challenges with avian mycoplasma. Mycoplasma gallisepticum primarily shows up as a respiratory type disease, and it is called chronic respiratory disease in chickens or infectious sinusitis in turkeys. And I have some pictures here that can show you how severe the infection can be. As I said, the prevalence of these avian mycoplasmas vary around the world, but every country where they're growing poultry has some challenges and has to deal with or think about how to control mycoplasma on some level. Next slide. Mycoplasma synoviae is the second most economically important of the avian mycoplasma species. It tends to be much more difficult to control than Mycoplasma gallisepticum, and we tend to have fewer tools also to control Mycoplasma synoviae. The prevalence of Mycoplasma synoviae also tends to be much higher than the prevalence of gallisepticum. And whereas gallisepticum shows up primarily as a respiratory disease, and you also have egg production losses, Mycoplasma synoviae tends to have a broader spectrum in terms of how it presents in the affected birds. So in addition to the respiratory disease, you tend to have inflammation in the joints, known as synovitis as well. that goes along with MS infection. Mycoplasma synoviae has also been shown to create eggshell defects. That's the thinning of the apical or the narrow end of the egg, as you can see in these pictures here. And because of the thin eggshells, these eggs are much more susceptible to cracking. So you have egg production losses that way as well. And you also have downgrading of eggs because of all of the mess that is created by these cracked eggs. As I said, Mycoplasma synoviae, you have a much broader spectrum in terms of what is presented and also degree of virulence. There are some countries where they have a very high prevalence of Mycoplasma synoviae, but they don't see a lot of clinical effect. Some, and quite often, the Mycoplasma synoviae infection only has subclinical effect, meaning the birds are not actively sick and dying, but you just have lower production. So both MG and MS can have subclinical effects in terms of just number of eggs that are being produced, decreased weight gain, increased losses of embryos. All of these different things can affect poultry production and affect profitability for the poultry industry. Next. Okay, so there are some unique things about Mycoplasma when we are thinking about how Mycoplasmas interact and or have an impact on the poultry industry. Most infectious disease can be transmitted horizontally. Can you next advance one more? So you think of your farm with your healthy, clean chickens, and there are many ways for infectious disease agents to enter or challenge that farm. For Mycoplasma, most of the issues with horizontal transmission are due to direct or indirect contact with infected poultry. And that tends to be non-commercial poultry. So small backyard farms or pet birds and pet chickens and those types of things. The prevalence of Mycoplasma in those populations tends to be much higher than that in the commercial industry. So most often what happens is that you have an employee or a farmer that has some kind of contact with infected birds, and then they bring Mycoplasma to the farm. Next slide. So in addition to horizontal transmission, the added impact of Mycoplasma is going to be because it is also vertically transmitted. So if you have hens that are infected with Mycoplasma, the Mycoplasma can be transmitted to their offspring through the egg. So when we are thinking about how to control Mycoplasma, we can't just think about keeping our farm clean in terms of biosecurity and trying to prevent infection that way. We also have to think about vertical transmission and how to stop or control that. Next. So the poultry industry is a pyramid. We tend to have down here at the bottom of the pyramid, which is the largest numbers of animals. Those tend to be the products that end up in the grocery store. So those will be the broilers. And then we also have commercial layers that produce the eggs that are down at the bottom of the pyramid where we have all of these large numbers of birds. But as we get higher up in the pyramid, the next level is called multipliers. Those are the parents of the commercial layers or the parents of the broilers. We tend to have fewer of those. And then up at the top, we have a few very expensive, very valuable birds that are pedigree stock. And you can imagine that if your pedigree stock is infected, then your entire pyramid is affected. So it is very important to focus Mycoplasma control at the middle and the top of the pyramid to make sure that we have adequate control of Mycoplasma because of that vertical transmission. Next. Okay, so how do we control mycoplasma? There's several different ways to do that, and I like to divide mycoplasma control options into two broad categories, short-term and long-term. So short-term are options that are available to you when you have an infected flock today. What are you going to do if you have responsibility for a mycoplasma positive farm? One, you can eliminate the flock. This is a test and slaughter approach that's been used for many decades in the US in particular. In the US, there's a very low prevalence of mycoplasma, and therefore we can afford to eliminate positive flocks if we choose to do so. In other countries, especially different parts of the world, the mycoplasma prevalence is so high that we can't afford to eliminate all of the positive flocks, because then we won't have any industry left if we eliminate everything that is positive. Quarantining and isolating are also a short-term option. We can stop the mycoplasma from spreading horizontally for a short time by quarantining or isolating, but eventually the mycoplasma is going to spread. Antibiotics, antimicrobials, any kind of medication is also considered in a short-term option. If the flock is positive, we can't eliminate them, then we need to treat. We need to medicate in order to prevent the effects of the infection from affecting that flock, but also to stop the horizontal and vertical transmission of mycoplasma from that flock. In the long term, biosecurity, of course, is important. We may or may not eliminate flocks as part of a long-term plan, but medication and vaccination are also considered to be long-term. Vaccination is not a short-term option. There is not a benefit to vaccinating flocks that are positive today, at least not a very large and measurable benefit. So vaccination is part of a long-term plan to control mycoplasma. And by introducing vaccination, we can change the prevalence of mycoplasma in a geographic area. So we can eventually move towards a prevalence where we may want to consider actually eliminating flocks one day. Next. So there's several good reasons to vaccinate birds for mycoplasma. As I said, it can control the clinical effects, so prevent clinical disease. It can prevent egg production losses. It can reduce egg transmission of our virulent field strains. And it can also reduce antibiotic usage. So I think we're all moving towards, globally, a more rational use of antibiotics everywhere. And being able to combine vaccination and targeted use of antibiotics, we are able to use antibiotics in a judicious way. Next. So in terms of vaccines or immunizing agents that have been used to control mycoplasma, there are inactivated oil emulsion bacterins. These are fairly simple to produce. We basically grow up a whole lot of mycoplasma, inactivated, and then we have an oil and water emulsion that acts as an adjuvant as well. These do work, they do have efficacy, but they are not as efficacious as the live vaccines. There are some recombinant vaccines. So those are usually a fowl pox vector that is expressing a few mycoplasma antigens as well to get an immune response to the mycoplasma. They are not very efficacious at all. They're less efficacious than the bacterins. And then we have live vaccines, which are, as I said, the first choice in terms of efficacy for a vaccine for mycoplasma. Next. So there are different kinds of live vaccines in terms of how they're produced and developed. Natural exposure is a form of vaccination. It is, of course, very crude. You basically just have no biosecurity and expose your birds and hope that they develop an immune response after that. naturally occurring avirulent strains, and then there are several ways to attenuate in vitro any virulent strains that may come about, so that you can end up with a vaccine that is attenuated and safe. Now, in my opinion, naturally occurring avirulent strains are the best option in terms of safety for the live vaccines. And as I said, the live vaccines are very efficacious. And the problem that we've had with them in the past is a safety issue. Sometimes they tend to revert to virulence, and the in vitro attenuated strains, especially when we use chemical mutagenesis or targeted mutagenesis, tend to be not as stable as something that is naturally avirulent anyway. So they tend to revert much more quickly and present a safety problem. Next. So there is no perfect vaccine, of course. There are pros and cons to each of the different vaccines and different things that have to be weighed. But I hope that we've made some headway so far in producing, hopefully, safe, efficacious vaccines for the poultry industry. And I look forward to continuing our partnership with ECO. Thank you.
Thank you very much, Naola. We're now going to hear from Natalie about the vaccine solution to poultry mycoplasmas. And Natalie will give us an update on the poultry mycoplasma vaccines that ECO is developing for the future. Over to you, Natalie.
Thanks, Hafid. Thanks, Naola, for setting the scene on the importance of mycoplasma in the poultry business. Next slide, please. Our new Mycoplasma synovial live vaccine will be commercialized under the name ECO Vaccine MS. This vaccine is an improved solution for an underserved market where there are very few registered products which are not available in all geography. As it was mentioned by David during his introduction, poultry will become the primary global meat source in the next 10 years and will play an important role in feeding the growing world population. As Naola was explaining, Mycoplasma infection have a huge economic impact globally for the poultry producer, reaching 640 million pounds per year. Mycoplasma is causing respiratory infection, a reduction of weight gain and egg production, chick and embryo mortalities, and carcass contamination. Some years ago, ECO decided to partner with the University of Georgia, and more specifically with Naola, to develop a best-in-class vaccine with superior efficacy and reliable supply to strengthen its leadership position as the Mycoplasma solution provider. Next slide, please. For ECO, it was important to develop a Mycoplasma synovial vaccine having superior key attributes. ECO decided to use a safe technology, as explained by Naola. The vaccine strain is a naturally attenuated US isolate with no genetic manipulation of the strain and no risk of reversion to virulence. The vaccine strain is safe when administered to young chicken at 4 weeks of age. The vaccine strain is also free of antimicrobial resistant genes. As I said in the previous slide, Mycoplasma synovial is causing different diseases. ECO is targeting 3 label claims for its new vaccine strain. First, protection against respiratory disease, what we are calling ursaculitis. Second, a protection against foot pad and joint inflammation, what we call synovitis. And finally, a protection against egg production loss. This is caused by ovarian regression after Mycoplasma synovial infection, where the follicles, or what we call egg yolk, degenerate instead of producing an egg. Finally, ECO is also aiming at providing a reliable supply of the vaccine, which will have more convenient storage attribute as the competitor product. Next slide, please. On this slide, I would like to expand on the 3 efficacy claims that ECO is targeting for ECO Vaccine MS. On the graph to the left, 52% of the placebo chickens show respiratory disease after challenge whereas only 11% of the bird vaccinated with EcoVaccine MS suffer from respiratory disease after challenge, which show really an excellent protection of the respiratory tract. On the graph to the right, 70% of the placebo birds show footpad lesion after challenge, whereas only 39% of the chicken vaccinated with EcoVaccine MS had footpad lesion after challenge. The lesions seen were also less severe in the vaccinated birds. On the graph at the bottom, young female birds were vaccinated and kept until they reached egg production. At 23 weeks of age, they were challenged and egg production was followed. 2 weeks post-challenge, there was a drastic drop in the egg production in the placebo, and this was associated with about 60% of the birds suffering from ovarian regression. In the vaccinated group, the egg production was not impacted, and none of the birds were suffering of ovarian regression. Next slide, please. These results are very encouraging, and the team will continue to work on completing the pivotal efficacy study. I think we are one slide too far. Thank you. These results are very encouraging, and the team will continue to work on completing the pivotal efficacy study as well as the duration of immunity study. As Mike will explain later in the presentation, he and his team will complete the technology transfer to our CDMO partner, which will be followed by the production of the consistency batches. And once we will have the GMP batches, we will perform the field safety trial in the US and in Europe. Then ECO will be able to complete the submission of all documentation to the USDA and to the other regulatory agency. Next slide, please. I would like to give a last word about the status of our USDA submission. Altogether, for both product, EcoVaccine MS and EcoVaccine MG, ECO submitted about 20 documents to the USDA so far, which included draft outline of production, development plan, master seed report, summary of information format, which is a kind of risk assessment of the product, and several protocol and report related to safety testing. We just submitted another pivotal efficacy protocol to the USDA. The next submission will be related to site information about our CDMO partner. We are currently working on transferring the manufacturing process and the assay to our CDMO partner, as Mike will explain in a few slides. When the consistency batches will be produced and the field trial completed, all documents will be submitted to the USDA to obtain the license in the US. Thank you very much for your attention.
Thank you very much, Natalie. We'll now hear from Brian on the innovations that he has been working on to deliver some breakthrough solutions to birth virus, but also other respiratory pathogens of importance in the swine industry. Over to you, Brian.
Thank you, Hafid. I'm happy to be here this morning to tell you about two of our exciting swine research projects. Start with the next slide, please. The first of those projects is EcoVaccine PCV2 MHP, which is a bivalent respiratory vaccine for pigs. Bivalent meaning two targets, a viral and a bacterial target in this case. Our viral component is porcine circovirus type 2, or PCV2. PCV2 leads to severe economic losses in the swine industry, estimated at over 489 million pounds per year in the EU, and over 50 million pounds per year in the UK alone. You heard earlier about the importance of Mycoplasma disease in poultry, and it does play an important role in disease in swine as well. Here, our bacterial component is Mycoplasma hypopneumoniae, or MHP. MHP causes reduction in production profits estimated at over 4 pounds per pig globally. Next slide. So why are we working on a PCV2 MHP combination vaccine? EcoVaccine PCV2 MHP will be the first swine vaccine delivered to the market by Eco Animal Health, and as such, it'll become the cornerstone of our swine biologics program. Many animal health companies do have a PCV2 or an MHP vaccine, but not all of them have what is called a ready-to-use or RTU combination vaccine. That just means all the components are in a single bottle, as opposed to being in separate bottles which need to be mixed or delivered separately to the pig. The goal of our vaccine is to be an RTU. Those commercial RTUs that are available, not all have great safety profiles, meaning they can be reactive after administration to pigs. Similarly, not all of the RTUs have great efficacy to either the PCV2 or MHP component. So this is where we feel Eco Animal Health has a great opportunity to step in and take market share in this area. Next slide. Here are some key attributes for our vaccine. For the PCV fraction, we're relying on proven technology by expressing what is called the capsid protein, which has been demonstrated to be highly efficacious as vaccine in pigs. Where we distinguish ourselves is we are using a PCV2 capsid sequence, which is unique in the industry and provides cross protection against multiple genotypes or strains of PCV2. Should be noted that when circovirus vaccines were first introduced to the market in the mid-2000s, PCV2A was the predominant strain, and as such, a lot of the commercial vaccines use the PCV2A sequence. Since that time, PCV2A has evolved to other genotypes, noted A, B, C, D, all the way to G and H, with today being PCV2D as the leading prevalent strain. For our MHP component, we're relying again on proven technology of fermentation with inactivating the whole culture of Mycoplasma hypopneumoniae. Where we distinguish ourselves is by using a recent European isolate of MHP. A lot of the commercial strain vaccines available today have strains which were isolated all the way back to the 1980s or even the 1960s. So we believe our more recent strain better reflects what is happening in the field today. For adjuvant, we're using an oil and a water emulsion technology, which has been proven effective in both animal health and human health. We add a little bit of a twist with the addition of what is called a copolymer, which acts as an immunostimulant. That just means it gives the pig an immune system boost. Next slide. You put all those components together in a bottle and administer it to a pig, and how does it perform? Here's an example of two clinical trials where we vaccinated and then challenged animals with either PCV2B on the left or PCV2D in the study on the right. The y-axis describes the total histopathology score, which means we just collected tissues at the end of the trial, looked at them under the microscope, and examined for signs of lesions or tissue damage. In the blue bar represented here is our eco vaccine candidate, which provides a statistically significant reduction of histopathology score compared to the black bar, the placebo-treated animals. We also included in this trial two commercial vaccines. One is an RTU containing both PCV2 and MHP. The other is a PCV2 only vaccine. And you can see our eco candidate performs very similarly to those commercial vaccines. A similar story in the figure on the right, again, where we challenged with the predominant field strain currently, PCV2D. In blue, our eco vaccine candidate provides for a statistically significant reduction of histopathology score compared to placebo-treated animals, and again, performing very similar to a commercial vaccine. Taken together, this also again demonstrates cross-protection to multiple genotypes of PCV2. Next slide. As a bivalent vaccine, we need to demonstrate efficacy against both the PCV2 and MHP components. Here is a vaccine challenge trial where pigs were challenged with MHP. As the name implies, Mycoplasma hypopneumoniae causes pneumonia in the lungs of pigs. At the end of the trial, we examined those lungs and determined the amount of lung area affected with pneumonia, and that is the score represented on the x-axis at the bottom of this figure. You'll see in blue again our eco vaccine candidate providing a statistically significant reduction of lung lesions compared to the placebo-treated pigs. We also included a commercial MHP vaccine in this trial. You can see no difference between the commercial vaccine and our non-optimized research vaccine here. No statistical or clinically relevant difference between those vaccines. We continue as a project to further optimize that MHP component, trying to improve yield and further improve our efficacy. Next slide. Looking ahead, we have an important milestone coming up for this project. We intend to have a stage gate transition moving from proof of concept into exploratory development. After that begins, we'll begin the production and testing of master seeds. All those are our frozen stocks of our PCV2 and MHP components, which are then tested and approved by regulatory agencies and is a key first step towards commercial production. As I mentioned, our optimization work with MHP that continues through the work of fermentation and scale up from lab scale, 1 liter or less, to 10 liters to 50 liters, working up towards commercial production scale, all the while maintaining consistent and robust growth. Our clinical program will remain active as we look to confirm the protective doses of our PCV and MHP component, as well as confirming the onset of immunity of our vaccine, meaning we'll determine how long after vaccination a pig is protected from infection. All this time, we'll also transfer this technology and methods to our target contract manufacturer so they can begin working towards being ready for commercial production. Next slide. So I'm going to transition away from the vaccine discussion for that first project into monoclonal antibody discussion for our second project. This is a monoclonal antibody towards porcine reproductive and respiratory syndrome virus, or PRRS as it is known. Now, PRRS is a devastating global disease in the swine industry, leading to annual losses estimated at 545 million pounds in the US and between 65,000 and 566,000 pounds per farm in the EU, highly dependent on the number of pigs and the type of production system on any given farm. Why is PRRS such a bad pathogen? PRRS has a very high transmission rate between pigs. There are also a high number of viral variations in the field. A pig that is infected with 2 or more strains of PRRS also has the opportunity for those strains to recombine, leading to even more viral variation. Control efforts for PRRS are mainly focused through the use of vaccines in the market. There are currently 2 types of vaccines available, a modified live or an inactivated or killed vaccine. Modified live vaccines have the downside of being able to spread to non-vaccinated animals as a live virus. And again, if present in the pig at the same time as a field strain, can still lead to recombination events. Both modified live inactivated vaccines have limited cross protection to the many field strains that are available as well. So we feel the industry is primed and ready for a disruptive and new approach towards the PRRS problem. Next slide. Here's an example of efficacy of our monoclonal antibody in a clinical trial where pigs were challenged with a European variant of PRRS. The figure on the left is viremia, and that just means the amount of live PRRS virus circulating in a pig. You can see pigs that were administered our monoclonal antibody have a statistically lower amount of viral load on days 2, 5, and 7. For rectal temperature, over the course of the challenge period of this study, there were statistically fewer observations of fever in monoclonal antibody treated pigs versus the placebo pigs. A reminder that one of the R's in PRRS stands for respiratory disease, and similar to the Mycoplasma disease I described earlier, PRRS does cause lung lesions in pigs. If those lesions affect greater than 15% of the total area of the lung, we consider that severe lung lesions. And in this trial, 1/3 of the placebo pigs had severe lung lesions versus none of the monoclonal antibody treated pigs. Last is body weight. Pigs administered our monoclonal antibody had statistically greater average daily weight gain than placebo treated animals. Next slide. Some upcoming goals for this project: we'll continue to work on improving the protective duration of our antibody, which means we'll continue refining to seek to keep the antibody circulating longer in the pig. That leads to a dose refinement, really, as the longer the antibody is circulating in the pig, the less amount of antibody that needs to be administered. Our lead candidates will continue assessments for developability, which just means we're exploring options on how to most cost effectively manufacture those antibodies. And lastly is our clinical trial listed here. I described clinical trial where we challenged pigs with a European variant of of PRRS, and we'd like to complement that trial by evaluating our antibodies' efficacy against North American variant of PRRS. I will note that we do have in vitro or lab data, which shows that our antibody can be and is effective against both of those strains. So we're optimistic going into that clinical trial next year. And with that, I thank you for your time and attention today.
Thank you very much, Ryan. We've always wanted an injectable antimicrobial to complement the Evlozin brand, and Alphonso will now share with you some exciting data on a novel long-acting injectable antimicrobial to treat swine respiratory disease with a single shot. Alphonso will also share with you data related to a nanobody innovation that we have been developing against necrotic enteritis in poultry. Over to you, Alphonso.
Thank you, Hafid, and thank you to all attendees that have interest in our work. I will start talking on pigs and this unique formulation that we are developing, and then we will move into chickens, as Hafid said, to discuss our alternative treatment for necrotic enteritis. Next, please. We've been talking about vaccines, and vaccines are an excellent tool to control diseases, but we know that in some situations, the treatment with antibiotics is necessary. And a good example is swine respiratory disease, which is a mixed bacterial infection that hits all the stages in pig production, causing tremendous losses to the industry worldwide. There are reports of several millions losses to the industry, and these are caused by poor performance of the pigs in places where disease is endemic, but also due to mortality in acute cases. So if pigs are not treated, they will suffer unnecessarily, or in the worst cases, they will die. At the moment, current treatments often imply multiple dosing, which is first not handy for the farmer because it is labor intensive, but it is also negative for the animal welfare of the pigs because they will get stressed with the several handling and several injections. To address this gap, we've partnered with a biotech company to develop a unique treatment that offers long protection with one single administration. Next, please. The beauty of this formulation is that using the thermosensitive gel that David described at the beginning of the presentation, we have developed a sustained release formulation that offers long duration of efficacy with one single administration. We've generated data that we will see in a moment that show that we have better protection against some relevant pathogens that other one-shot products in the market, and we will see it in a moment. We've talked also about the improved animal welfare because of the less handling, but in terms of safety, we also have data that show that this antimicrobial is a low risk of antimicrobial resistance development. Furthermore, this antimicrobial is not used in human medicine, and that makes it very attractive because there is no risk of transmitting resistance from the animals to human. This is a controlled release technology, and it is not a replacement of Avulocin. It's just a complement to increase the therapeutic cover, and it is an added tool to the toolbox to control swine respiratory disease that we were partially covering already with Avulocin. Next, please. I'm excited to show you some data for this unique formulation. The first thing we've done is to investigate how the drug behaves in a pig when we inject it. And we've done this in a pharmacokinetic study, and you can see the results on the left graph that show the plasma concentrations when we inject our formulation or when we give the same antibiotic with a competitor two-shot product. What is important in this graph is that if you look at the green line, which is our antibiotic, we stay above the yellow line for at least 5 days. This yellow line is very important because it indicates the concentration that stops bacterial growth. So we are able to stop bacterial growth and get therapeutic concentrations with one single administration for several days. That's the pharmacokinetic theory, but this also translates into efficacy in infected animals. We've tested the efficacy of this formulation in an experimental model, and we've done it using the hardest conditions possible, using the Actinobacillus pleuropneumoniae infection model, which is a mortality model. And you can see in this graph that represents pig survival during the study, that if you look at the blue line, that is the untreated animals, all animals died after 2 days post-infection. On the contrary, if you look at the green line, which is our formulation, 100% of our pigs made it to the end of the study. They survived. And in this study, we had a comparator group, a market leader, the treatment of swine respiratory disease, which is this orange line. And you can see that this market leader only saved the lives of 40–50% of the animals. So we've shown superiority in a very severe challenge model using a very relevant pathogen for the industry, because Actinobacillus pleuropneumoniae, APP, is a re-emerging problem for the farmers, in part due to the lack of effective vaccines. So next, please. Planning ahead of the registration and to address the regulatory hurdles that we know that will come for the registration of antibiotic. We've partnered with key opinion leaders in the field of antimicrobial resistance. And together with these experts, we have developed what we call the artificial pig, that you can see this, or those cables and bottles that you can see on the figure on the right-hand side. The fancy scientific name is the hollow fiber infection model, which is inspired in human medicine. And we call it artificial pig for easiness. And we have developed it for implementation in veterinary medicinal products. Basically, what we do in this infection, in this artificial pig, is to investigate what the drug or our formulation would do in pigs that are infected with certain bacteria. And without going into much detail, what we have seen using this assay, this testing, is that with our formulation, we can eradicate target pathogens. And what is also very important is that we have observed that we do not induce antimicrobial resistance in the bacteria that are present, which is very good in terms of safety for the products and will make the regulatory pathway easier. Next, please. In terms of upcoming goals, all going well, this project will be moved into full development soon. And once we enter full development, we will start the clinical program and run all the studies that are necessary for a registration. We have also developed the manufacturing process. And once we are in full development and in parallel to the clinical program, we will start scaling up the process to make a product ready for marketing. I'm very excited with having this added to the pipeline and to complement Avlucin. So with this, we jump into chickens, necrotic enteritis, and our novel approach for the control of necrotic enteritis, which is a disease caused by the toxins of Clostridium perfringens. Next, please. Necrotic enteritis has been a huge problem for the industry, for the poultry industry during several years, but especially in the last years, it is gaining more and more importance, partially because there is a ban on the use of antimicrobials as growth promoters. There are reports that indicate that the losses for the industry are several billions around the world, and these losses are due to the poor performance of birds that are suffering the subclinical form of the disease, but also because of the losses that due to mortality when there is a more acute presentation of necrotic enteritis. Farmers nowadays are under a lot of pressure because they are banning all the tools that they have to produce healthy animals. And at the same time, they are not being offered sufficient alternatives. And that's where ECO is committed to give our meat producers sufficient alternatives to control all the diseases and deal with their daily problems. Next, please. The mode of action, or the beauty of this product, is that it is an antibody that is that targets the toxins that Clostridium perfringens produces. These toxins damage the gut wall and result in lesions that impair productivity and also may cause mortality in more acute cases. So by inhibiting these toxins, we reduce the severity of these lesions, and as an end result, we have birds that grow better, and we will also reduce the mortality. We've put a lot of thinking in making a product, on developing a product that is easy to administer. That's very important for chickens. And we have successfully demonstrated that we can administer this product either in feed or in water through the oral route, which is very convenient for the farmers. And it is also, as I mentioned before, for the antibiotic, it is also good if you don't need to handle animals several times because of the animal welfare. This is an innovative approach using the nanobody technology. And what is very attractive from this concept is that we target the toxins, not the bacteria. Therefore, there is no risk of antimicrobial resistance development. Next, please. So with this, I'm very, very excited about the data that we've generated this year with these antibodies. The first thing that we've done is to test the potency in vitro. So basically what we do is to mimic in a plate what the toxins would do in an animal. And we study the effect of our antibodies on those toxins in vitro. What the graph on the left side is telling is that at nanomolar concentrations, we inhibit the damage that the toxins are causing. And nanomolar potency for an antibody is an indication of high potency. So we have very good candidates. And we've brought these candidates into infected animals. So the right-hand side, you have the data from a challenge study in which we infected animals with Clostridium perfringens that produce the toxins that cause necrotic enteritis. And we treated our birds with these antibodies given orally in water. And what we see in this graph that represents the PEM mortality is that our antibodies did have an effect on mortality when given orally. These data are very relevant to me because this was a very, very severe experimental model with mortality up to 90%, which is unusual for necrotic enteritis. And yet we saw an effect of our antibodies, and this was our first attempt with these antibodies. So from now on, it only can get better. Very excited about these results. Next, please. With regards to the upcoming goals for these projects, we are exploring alternative methods of delivery and administration routes. And one of the things that we are investigating at the moment is a novel delivery method using a vector, bacterial vector, that would bring the antibodies to the target site of action. That will help us to get a more efficient dosing, and ultimately this will translate into lower cost of goods. As I said, in parallel, we are also investigating other administration options. And once we have defined the final product, we will be moving into transition to exploratory development to fine-tune the dosing and to start the developing the manufacturing process. I'm very excited about this technology because it's a game changer for the treatment of enteric diseases. We're focused now in poultry, but it is a technology that can be extended to other species. And with this, I would like to thank you for your attention, and I'll be open for questions at the end.
Thank you, Alfonso. Scaling up production and manufacturing high-quality vaccines and biologics is a key component of what we do. And Mike will now share with you how we're addressing that critical piece of the development cycle, focusing on the late-stage development pipeline. Over to you, Mike.
Thank you, Hafid. I'm also delighted to present to you an update on ECO's biological manufacturing network strategy to deliver our exciting and diverse portfolio as highlighted by my colleagues. The biological manufacturing network strategy consists really of 3 phases. The first phase was a short-term strategy to collaborate with the US CDMO, a contract development and manufacturing organization, to initiate an ECO-led co-development program for our portfolio, while informing the U.S. Department of Agriculture on our U.S., Canada, and Latin America licensing strategy. The first phase was successful in accomplishing these goals. However, during this collaboration, we were informed by outside sources that our CDMO could potentially be up for sale. This was a critical turning point in the future of our portfolio. We met with the ECO leadership team and proactively initiated phase 2 of our strategy by securing a European CDMO and informing the USDA of our intentions to establish a US quarantine site. Now, this allows for importation of product as a permittee for sale in the US and other markets except China. In parallel, we needed to secure our US development operations to ensure a smooth transition. ECO aggressively moved to build a research and development lab in Des Moines, Iowa, to efficiently transition development and all regulatory control away from our US CDMO. The US CDMO was sold on April 3rd of 2023 to the Mars Corporation, and we amicably transitioned all regulatory control and development activities to our new ECO facility, and were fully operational by the end of April of 2023. No small task. These well-planned decisions have now put us in a position of more control of our R&D development, regulatory interactions, and strengthen our brand recognition as we launch products in the US and other countries that will accept our US dossier. This foresight and ability to execute quickly prevented a potential 1 to 2 year delay due to legal, regulatory, and developmental activities. Lastly, the ability to consolidate tech transfer and production of all markets except China in collaboration with our European CDMO also provides an opportunity for improvement in cost of goods, as well as due to economy of scale and future margin improvements as the products and new markets mature. The last phase of our manufacturing strategy is the China for China initiative. We have signed a memorandum of understanding with our current China partner, which is key to securing an R&D lab as well as a cGMP pilot facility for in-country R&D and clinical support. In combination with our US-based R&D lab, we'll be able to quickly leverage our knowledge transfer to China to ensure the fastest approach to licensure of late-stage products, which includes 2 poultry and 1 swine product. This phased approach will build a foundation for a cost-effective transfer to a global CDMO production facility to deliver on our diverse portfolio of vaccines and biotherapies. It could also serve as a potential toll manufacturer for EU companies if so desired. Next slide, please. This slide conveys how we're streamlining our new assets to quickly promote lead candidates from proof of concept all the way through to full development. Our multidisciplinary project team will continue to utilize our strong CRO development collaborations for emerging solutions as discussed by Hafid, as well as our specialist CDMOs to find the best production strategy with our in-house R&D experts for efficient technology transfer to our global CDMO partners. Hafid, could you please advance the lab animation? The ECO R&D Lab also provides a key hands-on analytical and process development approach to expedite the knowledge transfer to our CDMO as well as to our U.S. clinical and regulatory teams. This lab is critical in providing clinical study vaccines and analytical support to fulfill licensing studies in a timely fashion. Our commercial and manufacturing team will continue to evaluate the most cost-effective strategies to minimize costs and deliver quality products to our customers in a timely fashion. As you can see, we have already, in a short period of time, adapted to unexpected changes, consolidated, and provided work streams for delivering products to all global markets, including China. Next slide, please. This last slide illustrates our developmental flow for EcoVax and MS technology transfer to our European CDMO, as highlighted by Natalie. This consists of really 6 phases of the tech transfer process: materials, measurements, methods, machines, environment, and personnel, which all must be firmly orchestrated by our multidisciplinary project team to ensure timely delivery of key steps in the tech transfer process to our CDMO. The green highlights the complex activities that have already been completed to build a future production platform for our range of products. The blue highlight the ongoing activities to prepare equipment, people, testing labs, and production areas for the final analytical and process tech transfer activities, leading to the large-scale production of consistency batches for product registration and launch in the US and in Europe. The black highlights tasks currently being planned to finalize the technology transfer in order to deliver clinical materials to support the field safety studies for the final regulatory approval of the product. To conclude, we feel confident in our global teams, our R&D lab assets, and our current collaboration to deliver on our aspirational biological manufacturing network strategy for the delivery of our diverse portfolio in the years to come. Thank you.
Thank you very much, Mike. We'll return now to the key slide on the value of the pipeline. I hope the presentations that you've seen and heard from the team have given you an insight into our results and data with the mid and late stage pipeline, and also into our strategy on how we plan to manufacture those products to supply our markets. From what you can see here, our near-term mission and deliverables over the next 3 years are going to be 3 new product approvals: US approval of EcoVaccine MS and EcoVaccine MG by the first half of 2025, and the European approval of our injectable long-acting antimicrobial by the end of 2026. I believe we have an exciting R&D portfolio, and we look forward with optimism to turn it into new products that will broaden and diversify our offering to the swine and poultry producers. And I'll now hand over to Chris for a financial perspective on the pipeline.
Great. Thank you very much indeed, Hafid, and thank you, colleagues. It's been a fascinating presentation. I've been in exalted company this afternoon. I'm the first and the only speaker who hasn't got a PhD, but nevertheless, we'll have a go at providing some financial information to complement what we've heard. Clearly, we've heard a lot about science this afternoon. It's been a pretty fascinating set of presentations. Hopefully, certainly in my mind, it's brought some of that science into perspective and given a bit of the sort of commercial background to why we're doing it and what the potential is. So let's turn that into financials. I think one of the first points to note is we're looking at a portfolio here. We've had a deep dive into half a dozen or so of the projects, but there is a lot more within the portfolio, which is early stage. We've only looked at the clinical stage projects at this in this presentation. And when you look at that portfolio, like any good portfolio, it's got a mix of risk and reward, and it's also got a mix of projects at various stages. So some which are quite close to market, others which are still in the lab. All projects, regardless of size or stage of development, are subject to rigorous business modeling. Right at the beginning, we build a business model, and we refine that business model and revisit it at each stage of transition through the development program. The models are built from the bottom up. So we start with animal statistics, so numbers of animals in production. We then pair that statistic down into a component which are medicalized, a component which are treated, component which are diseased, and then finally we have a look at our likely market share given what we're offering compared with whatever the competition might be in the marketplace. So that is how we derive revenue. We also, from that revenue number, look at costs of goods, and particularly with vaccines, cost of goods is heavily dependent upon volume. So we model the cost of goods dependent upon the volume and the scale up into peak revenue. Also, we model a phased entry into the various marketplaces that we're going to tackle. As you know, we address the swine and poultry markets around the world, and each market will have its own regulatory environment and regulatory requirements. And we tend to do those in series rather than in parallel. So clearly that implies a phased entry by market. We also look at what the incremental costs might be for getting into some of these products. And the key thing to note here is that the route to market for all of these products will be through our existing channels. We're looking at the same species, and we're looking at the same distribution models. Therefore, the customer base and the route to market is the same. The result is that the incremental costs of entering a market are low. And nevertheless, we do program in some incremental costs into our models. Of course, we shouldn't have to say this, but it is important to emphasize that at any stage gate, we will only look at the future opportunity and we will disregard sunk cost. It's a human trait that one finds it hard to disregard costs that have gone before when considering whether to proceed. But in our case, we are very firm about that particular point. Ultimately, I'll be presenting a bit of a valuation which suggests the net present value and IRR for these projects. So first of all, a table which essentially summarizes part of the answer. And the part of the answer that I want to present right up front is peak revenue. So that's the column two from the end. And this is the projects that we've heard about this afternoon, together with a row which is called others. That's the rest of the portfolio. And as a combination, this portfolio has a potential to deliver over £300 million of peak revenue, and translating into over £160 of EBITDA or profit contribution to the company. Clearly transformational. The R&D costs, which is the first column, are relatively modest for that kind of return. Also on the slide, you'll see the probability of PTRS, which Hafid talked about earlier on. And I'll talk again about PTRS in a moment. And then finally, there's the sales starting year. You'll see that I've arranged this group of projects in order of market introduction. That's first market introduction, not peak market, but first market. And you'll see what correlates is between the euro introduction and the PTRS. In other words, the probability of success measure that we're putting up here has a strong time component. And that is important to bear in mind. It's a slight misnomer calling it probability of success because that implies confidence. We are confident at everything we do, otherwise we wouldn't do it. But nevertheless, the way we compute probability is to factor in the risk around work still to be done. So how does that translate into into revenue? So on this graph, we've got 10-year revenue. In blue is the existing business, which is dominated by Avison. We expect Avison to continue to grow. It continues to gain market share in all the markets it operates in. And of course, it's supported by the demographics, which David talked about at the beginning of this presentation. Nevertheless, you can see the transformational effect of these new products, the blue line being the revenue derived from the products as they're introduced, and the combination being the yellow or gold line, which moves the revenue up to a factor of 4 times where we are today. And what that does to our profitability is a similar line. It's a near exponential growth in profitability. So moving on to some other modeling assumptions. I've talked about the bottom-up build of revenue. And I've also talked about the regional rollout or the phased rollout. Here it is graphically. And in this case, we've used this phased approach, which we are targeting for MG and MS, and used it for all of the products, which essentially means we're moving into USA and Canada first, followed by certain countries in Latin America a year later. Brazil and Mexico will follow a year after that, together with certain Southeast Asian countries, which tend to go hand in glove, and then finally going to China and Europe. So it's that sort of phased approach over 3 years into all marketplaces that we operate in. Now, probability of technical and regulatory success, we talked about that earlier. It is the product of the probability of being promoted to final development or the final stage of development, multiplied by regulatory risk. And that is why we get to the number we get to. It includes all of that. We had to assume some build up to peak revenue, and we've assumed that will be reached after 3 years of market entrance. So each market will build up over 3 years to its peak revenue, and then you get phased entry to each market after that. So for a given product, peak revenue will be achieved after possibly 5 years. We then have peak revenue remaining at peak for a plateau of 5 years, modeling it down to a taper of 10% of that peak by year 15. There's nothing in the current portfolio or in the history of Avison which suggests that tapering should happen, but nevertheless, the textbook of economic theory tells you that products do tend to wane over time, and that's the way we modeled it. Weighted average cost of capital, we've used 10%. We can debate whether that's too high or too low, but you'll see that I present the cost, the return on the developments in the form of an IRR as well, so that removes the question of what a particular entity's weighted average cost of capital is. Commercializing the products in China, we're going to use exactly the same structure as we've currently got. I'm not assuming in the model any change of equity. And that's notwithstanding that we actually do have a principle agreed that as we inject new IP into the joint venture in China, we will increase our equity share in that joint venture. We've included some incremental costs, which I spoke about earlier, and we've included some pre-launch costs. So getting into each of the half dozen projects or so that we looked at earlier, I present here in the same format each project, showing the addressable market size, the PQO market share, which in case of MG and MS, they're markets which are over 40 million pounds each. And we think we can get to a market share on average across all world markets of 37% and 25% in each case, with a cost to go in terms of R&D of just around about 5 million pounds each. Very high probability of success, reflecting the late stage of these projects and the proximity of commercialization, leading to peak revenue of over 11 million pounds, in case of the MG, 15 million pounds, and an EBITDA contribution to profits of 8 million and 7 million. So very healthy profit contribution in each case, and the net present value measured over the full life scale of the two products of approaching 20 million in each case. Mentioned IRR earlier, so this is a different way of looking at the return, and in each case greater than 40% here in a payback of 6 years. The next pair of projects we looked at, which are for the swine biologicals, so the PCV2 MHP combination, and also the PRRS virus monoclonal, much bigger marketplaces. So the addressable market size for these around 300 million pounds, and we think we can get to quite a nice market share in each case, 12%, 21%. These projects have got a fair way to go, reflected in the PTRS, but nevertheless, the actual R&D cost in the case of the PCV2 MHP is fairly modest. The monoclonal for PRRS virus is more expensive, as you would expect. This is a highly novel technology, highly groundbreaking technology. These are two of the most valuable projects we've got in the portfolio, and as a result, peak annual revenue is high, and the contribution to profit in the case of the PCV2 combo 18 million and nearly 40 million for the PRRS virus development, and very high net present value as well. So then we heard about the novel antimicrobial, which is the injectable antibiotic. We heard about that from Alphonso. A good market size of 69 million pounds with a 9% expected market share. This is a product which we do not expect to introduce in the US, and we've modeled it to exclude the US. So this is European and Far East markets in the main, together with China. About an R&D cost of about 3 million pounds to go, so not too far to go on this, albeit that we've modeled a fairly cautious PTRS on this, reflecting that combination of technical risk and regulatory risk. Peak annual revenue, 6 million pounds, but note that profitability is quite high. That comes directly from strong gross margins, and so a very useful contribution to our overall profitability for the group, with a net present value of about 7 million pounds. The other project Alphonso talked about was the enteric disease prevention product, which is the necrotic enteritis. Addressable market size 148 million pounds. I think that really reflects the impact that this particular disease complex has on poultry production. We think we can get to a 25% market share with our product, and it'll cost just about 8 million pounds to get there. Still early stage, reflecting PTRS. Of course, with that kind of market size, the potential for us is quite high, and you can see the revenue growth chart. Clearly, in the 10 years that we're presenting here, we haven't yet got to peak revenue there. You can see it's carrying on beyond the chart size. So this is one that's still a way out. So moving on to the final slide, which is trying to pull this together into some sort of valuation. First of all, let's look at the revenues and EBITDA again in tabular format. So we saw that early on. The net present values, which I presented as part of the individual projects, all add up to £330 million, including the rest of the portfolio, which we haven't talked about here, which is earlier stage. Now, one approach to risking or applying the risk factor to this present value is to take the PTRS, which we saw earlier, and multiply it in each case against the NPV. And that gives you a probabilized NPV. Sum that up and you get to £86 million. It's one approach. There are several approaches to risk factoring the portfolio. Another might be to look at our historic rate of attrition. And that statistic, since we started the R&D program back in 2017, it's been about a third. So another way that you could risk this portfolio is to say, well, a third of the projects will fall away. Nevertheless, what you end up with is a high proportion of that net present value coming through into a risk-adjusted number. The internal rate of returns in each case you see are quite high, and certainly well in excess of what the group's cost of capital is, whichever way you look at it. That's all very good. How affordable is it? Well, we model the cash flows for developing all of these programs through to completion, and you'll see here that the net cash generated, which is the turquoise or teal bar, is at least neutral in each case, and then turns positive from 2026. And that increases the cash balances as we go through the program. In other words, the program is funded from internal cash resources, not needing any external influence. So having a go at suggesting what our share price undervalue is this final table. Today, we're around the 106, 110 share price. And there's 68 million shares in issue. Now, if you take that net present value and you say, well, that represents the current value of all future cash flows, then that should, in a perfect market, be reflected in the share price. So therefore, the share price should be greater than it is by anywhere between £1.25 and £4. Several other ways you could look at this, including looking at the current ratio of share price to earnings and applying our new EBITDA as we progress through this program to that, and that gives you a suggestion of a share price in 5 and 10 years' time. You could look at EPS. There's several other ways of valuing up the stock, and I'm sure the audience will be well versed to do that, and I'll leave those thoughts with you. So I'd like to hand back to David, who will just round us out.
Thank you very much, Chris. Superb insight into our portfolio and the valuation. Just to remind you, we have shown you a handful of our assets in R&D. We have a broad and deep R&D portfolio. And just to bring this home, these are the 6 that we have highlighted today. There are earlier assets as well. But since we did this CMD early in 2022, the assets have moved further into the clinical stage and further into the late stage. So we have a rich and deep portfolio and a maturing portfolio that will exit into the commercial market in the near-term horizon. So a final summary I would like to say is that you can say that ECO is pregnant with innovation. We have more innovation in the company than we've had in our history. That innovation is both of high quantity and of high value that you've seen from Chris. And if you take even just a conservative estimate of that value, that's that conservative estimate could deliver over 200 million sterling in revenue and over 90 million pounds at the EBITDA level. So this would represent a substantial increase in the value of the enterprise, and in fact at that level is truly transformative, doubling and trebling in a near-term, mid-term horizon. So I hope you found this insights into the science, into the market, into the background, why we believe we have some great innovation in the pipeline. I hope you've followed the valuation and can see where we've drawn those numbers. I will now pass it back to Hannah to MC any questions that might come from the audience. Thank you very much.
How do you see the opportunity for your PRRS antibody and vaccines in the context of the gene-edited pigs that Genus intend to bring to the market over the coming years? Do you believe there is ample room for both to coexist?
Yeah, great question. Almost answered within the question, actually. We believe that there is such a large challenge of PRRS in the marketplace that there is more than enough room for several solutions. In fact, there is the need for several solutions. We think that that will give us and Genus and other vaccine manufacturers more than enough space to play with. Genus have still got some work to do, as we do, but we think that there's more than enough space for several solutions.
Thank you. Given the large crossover between antibiotic usage in PRRS positive pigs, do you have any concerns that either could cannibalize your existing franchise with Avicin?
We've modeled a small amount of deterioration of Avicin over the planning horizon. That is more to do with competition from the outside than cannibalization competition from the inside. Again, it's a similar answer in a way to the last one. There is space for both prevention for PRRS, and you also need treatment. So we perfectly believe that Avicin and our monoclonal can coexist together, but we have modeled a small amount of Avicin deterioration over the planning horizon of 10 years.
Okay, we're going to continue on a theme. Just on the commercial perspective for PRRS, the pitch to sell a preventative measure is presumably slightly different to that to a treatment such as an antibiotic. Can you remind us what your sales synergies you see between Avicin and your pipeline assets, which seem heavily weighted towards prevention?
Yeah, good question again. The synergies are around this. It's the same customer, it's the same route to market. We know the customer, and actually providing several tools for the overall control of challenging diseases is valuable. So prevention is about 1/3 of the market in the veterinary market through vaccines at the moment, depending on which species you're in, higher in some than others. But for the customer, being able to provide a preventative solution and a therapeutic treatment solution, which will still be needed, because not everything is 100%, not every animal may get vaccinated or prevented correctly or perfectly. So having both provides a degree of synergy. Having the same route to market and the same customer provides some efficiency and also some synergy. And many other companies do the same as well. So this is not a unique having both preventatives and therapeutics. That is a good approach, and we would love, and that's why we would love to enter the preventative side of the market, and that is what we aim to do.
Thank you. You mentioned cost. What is the total cost of getting the PRRS monoclonal to market? Do you need or would you want to partner this?
So the total cost was there in the model was around about 8-9 million. That's fundable and affordable from revenue, we believe. To the broader question, are we open to partnership? The short answer is yes, if it's right. We have had some approaches, not for that particular one, but for other assets, which we look at each one. Sometimes those partnerships or approaches are because the pursuer doesn't have a solution in their portfolio, or they're looking for a better solution, or they're intrigued by the technology. All of those are, if they are the right partner, are potential valid routes, and we would look at each of one their own. I think the summary is that we can do it ourselves, but we're not averse at all to a partner that could help us either accelerate commercially or accelerate our route to market, our speed to market.
Clear, thank you. Are you aware of any other PRRS monoclonals in late stage development that are closer to the market than you are?
I am not aware. Hafid? No,
no.
we're not aware.
Are there any exotoxin targeting therapies on the market already?
I think that relates somewhat to the necrotic enteritis. And again, I'll ask Alphonso to comment, but to my knowledge, no. Alphonso, do you want to elaborate
Yes.
or correct me?
Yes, sorry, I'm trying to switch up my camera. There are no products that I'm aware of that target the toxins right away. There are some, but there is one commercial vaccine that I know in South America, but it uses Salmonella genetically modified organism, which is not very desired from a human perspective, because it is Salmonella is a critical pathogen for human as well. But there are no drugs targeting the toxins right away.
Thank you, Alphonso.
Was the PRRS monoclonal internally developed?
Yes, with partnerships and collaborations, but the idea was internal, and we brought together two main partners to work on the idea and to turn it into a concept that we took into the pigs. It's been going on since 2018, so it's been a long-running discovery program. We had to screen a number of candidates to arrive to the one that Ryan has presented to you, and we hope to get it to the market mid-2027.
Thank you. The competitive threats to Avlacin, are they a risk of undermining the cash flow that you are relying on in order to remain fully funded?
Well, we are adept to managing competition. Avlacin's faced competition since day one, and there have been a number of generics on the market for quite a number of years, including in China and perhaps mostly in China. So we recognize that. We're adept to fighting that. We fight it in a number of ways by having good commercial expertise, having great technical services, good key account management, in the future providing a broader portfolio of solutions. So all of these things, plus producing quality products at the right time, at the right price point, which is not always the case in terms of quality with the generics, although there are some good quality ones around. So we face that. Avlacin has continued to grow in the face of that competition. But as I said, for prudence, we have at the far end of our planning horizon, we have planned for a somewhat lower Avlacin revenue. In the near term, that revenue is continuing to increase, but as we get to the more mature and further out years, that does we have modelled a decline in that. So we think, in summary, Avlacin will continue to provide a good and robust revenue. We can fund our R&D from our overall commercial activities. We do have access to capital as well, and we are open to partnerships, but we can plough this furrow alone with the portfolio that we have. But as I say, we are open to partnerships as well. So I hope that's a full answer.
Certainly, thank you. Do I gather that virtually all R&D expenses are in US dollars?
They're not. Well, they're not entirely in US dollars. There's a good proportion. Perhaps Chris, you want to elaborate?
Yeah, and we tend to budget in US dollars just because there is a large component of the R&D team in the US, and clearly we've got our lab in the US, so there's a sort of a natural affinity for the US dollar. But you know, you've heard from our speakers. Clearly, there were a number of European-based speakers there as well. And we do a lot of R&D here in the UK. We've announced the various collaboration partners that we collaborate with, Fulbright Institute being one, and the Vaccine Group being another, and Rosalind as well. These are all UK-based institutions.
And we take advantage of the R&D tax credits that are available to us in the UK with our R&D spending in the UK. So I think the short answer to your question is no, it's not all in US dollars. A good proportion is there's a degree in euros and a substantial amount in the UK in sterling as well.
Thanks. To what extent are the wider industry aware of the detail of what is in your pipeline?
Well, we're going a bit more public here, aren't we? So I suppose they're open to seeing this. We have not shared any further information than you have seen today with any partner or anybody in the industry. So what you are seeing today is as much as is publicly or privately known by other animal health players.
Thank you. In the US, do you need to get approvals from the individual states, or does the federal USDA approval suffice?
Federal US, let's not call it federal, but the USDA approval is a national approval for the US.
Thank you. Well, unless any more pop up, that is it for our questions. So that only leaves me to thank the audience for attending and for our many presenters for a very informative afternoon. Thank you all.
Thank you for hosting, Hannah. Thank you very much, everybody who attended.
Thank you.